This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 105, recorded on October 28th, 2010. Hey everybody, I'm Vincent Racaniello, and it's time for TWiV. And again, we have a complete, spelled C-O-M-P-L-E-A-T, crew, beginning with Dickson Despommier.
Hey, Vince.
They have that book, The Complete Angler.
We
right?
do, yes. I have that book, by the way.
Why is it spelled that way? Is it
It's an Old English spelling.
Old English?
Yes.
The Complete TWiV.
That's right.
How you been?
Been very well.
Haven't seen you in ages, right?
Oh, yeah, like since last week. Earlier today, actually, Vince, if you want to tell the exact truth.
It's two weeks in a row now. This is just unprecedented.
It could even happen next week, too. Who knows?
And we also have in Western Massachusetts, Alan Dove.
Good to be back.
Always.
Yes, absolutely.
Excellent. Good to have you as well. And then down the coast, North Central Florida, I always forget, is Rich Condit.
Howdy. How's everybody?
Everybody's fine.
We are all well and ready to speak viruses. It's an odd day. It's Thursday instead of Friday.
That's right.
Yeah, that's a little strange. Doing TWiV on a Thursday almost makes it feel like a Friday.
Indeed.
And it's not.
I tell you what, we're all going to take tomorrow off. How's that?
Well, Rich has to work. That's why he
Oh, darn.
can't do TWiV tomorrow.
Too bad.
He has some stuff to do. But we have cool stories today, as usual. You would think that following up last week would be tough, but it isn't in virology. It's always easy to find story. And the first one is something that we all have talked about, eradication of a viral disease. We've talked about eradication of smallpox. We've talked about trying to eradicate polio. And apparently, Rinderpest is about to be eradicated. I don't think we've ever talked about Rinderpest, right?
We have at least mentioned it briefly. I forget what the context was, but we've talked about it before. Okay. I know that because I didn't know anything about Rinderpest until we talked about it on TWiV. All my information comes from TWiV.
Me too, by the way.
Not a bad source.
Not a bad source at all.
It's up to date.
That's right.
It is vetted.
And when it's wrong, we admit it.
Well, people tell us.
That's right.
Too, so it's self-correcting, just like science.
Yep.
Cattle plague in German, right? Rinderpest. Schmutzdecke Rinderpest. I said two words in German right next to each other.
Das ist nicht recht.
Well, all our German listeners are cringing.
Yes.
Sorry, but Rinderpest is a cattle pathogen. Dixon, do you know about Rinderpest?
I do, actually, because it's the plague of sub-Saharan Africa. At least it used to be.
Yeah, so it's a food issue, right? Because it makes the
issue.
cows sick and
That's
the cows you need to eat, right?
Correct.
In fact, the first article I came across is from the Food and Agriculture Organization of the United Nations.
Or otherwise known as FAO.
FAO. And of course, for them, it's a global food issue.
Sure.
So it's been a global effort to eradicate this virus. So let's do a little background on Rinderpest. Rinderpest is a virus related to measles virus.
Uh-huh.
It's the same family. And it is a negative-stranded RNA virus.
One of the 7 ways that viruses
Dickson.
can reproduce.
Wonderful.
According to Dr. Baltimore, of course.
That's right. With an envelope.
Yep.
And that's about all we need to know there. It doesn't infect people, even though it's in the same family as measles.
By the way, just as an aside, Alan, I think I've thought of the title for this episode. It says, "May I have the envelope, please?"
Oh, very nice. So,
Good.
are you here every night?
Only on Wednesdays, and try the veal, please.
May I have the envelope,
Now that
please?
you can try the veal because
That's
the
very good.
Rinderpest is gone.
Well, Rinderpest makes cattle very sick, and apparently it's on the edge of elimination. So this FAO article says it's almost eradicated. Next year, we're going to declare it eradicated.
Wow.
Okay.
That's brave.
So I guess it sort of is already scientifically. We'll go over some of the information. And all the press is saying it's eradicated.
Well,
But they haven't officially announced it yet.
right.
So this is a virus that's been around for hundreds and hundreds of years. I mean, way back in the 1800s, this caused very bad devastation of disease. And I found a wonderful article Well, there are two good articles. There's one in the Merck Veterinary Manual.
Right.
Disease of cloven hoofed animals
Mhm.
with fever, necrotic stomatitis. That's lesions in your mouth. Gastroenteritis, lymphoid necrosis, and high mortality. The most lethal plague known in cattle. Cattle are susceptible. Then a bunch of wild animals, buffalo, giraffes, a few others here.
Well, this is a problem then, isn't it, for elimination?
Well, they say it doesn't really persist in wild animals. And I think they've been surveying and looking for it.
Is this a vector-borne infection?
It's not vector-borne, so it's transmitted by direct contact among cattle.
That's the secret then.
By respiratory secretions.
Yeah.
So it's lethal, and then, of course, you can't harvest those cattle.
Nope.
And if it starts out when they're young, they never grow up and so forth. So that's clear why it could be a problem, right? Mm-hmm. And in fact, historically, as we said, it's been around for hundreds and hundreds of years, and there have always been epidemics. 1889, cattle shipped from India carried the virus to Africa. So that's when it went to Africa. And it started infecting cows there. And then wild buffalo, giraffe, wildebeests were infected. Loss of plow animals. Mass starvation as a consequence. So we can understand why there's been a major effort to.
So let me go back here
eradicate
a minute. Did you say it's primarily a disease of domestic cattle?
Yeah.
It says that it infects a bunch of.
Yeah, it does infect wild animals, but there's
it doesn't
no reservoir.
devastate them. It doesn't devastate them like it does domesticated animals.
Well, it doesn't. Apparently, they don't keep it. I don't know what the mortality rate is among these various species, but I gather that there's not nearly as much transmission.
Right.
It says here that cattle are the most important maintenance hosts. Sheep and goats are relatively unimportant. Among wildlife, African buffalo is the most important. Although there is some disagreement on the length of time the virus can persist in this population, some studies suggest that it disappears after 3 years.
There's that 3-year thing again. 3-year cycles.
Interesting.
So,
sounds like
it sounds like a disease of sort of domesticated or civilization, right? High enough concentration.
Yeah, that's right. Monocultures. Monocultures.
Yeah.
But, gnus are definitely considered a monoculture in Africa, so maybe they're also susceptible to this.
Because when I was considering this, the idea of eradicating a disease that has, that infects wild animals, as same as you noticed, Dickson, strikes me as a very difficult project. But I guess
and gnus can really spread fast.
That's right.
Get it?
I got it.
Okay. It says here, I mean, yes, this is the key, that if there were a reservoir in animals, you could never eradicate it. That's why you can't eradicate influenza virus, right?
And it also says in your article here that it's never established itself in North or Central America, the Caribbean, South America, Australia, and New Zealand. That's interesting.
Yeah.
Yes.
I wonder how it managed to do that.
I mean, that's why we can eradicate polio in theory, because there are no animal reservoirs. Clearly
it is actually really interesting that it did not manage to spread to. I can kind of understand maybe North America, temperate climate, it might not do as well, but why does this not? Why do we not have this in Central and South America?
Good question, because there probably is traffic among the animals.
I mean,
it's been around for hundreds of years.
Yeah, it's been around for hundreds of years. I'd be very, very impressed if we've simply maintained quarantine that thoroughly. That's possible that it just hasn't made the leap, but the other possibility is that it's simply been eradicated from those areas already.
Yeah, and the other thing is that the animal gets sick pretty rapidly, the domestic animals at least, and so therefore they wouldn't be shipped because they're not susceptible.
Right.
They're not likely to get on a boat.
Yeah, they mentioned that there aren't any carrier states where you have infection without disease, so you can tell when they're infected.
Yep.
Okay.
It says here also.
And I guess vaccination is much more common and easier to accomplish in most of the places you just listed.
That could be also,
Yeah, because we've also wiped out a lot of other cattle diseases in this country. I mean, foot and mouth disease, screwworm.
Yep. At least in Florida.
Which one in Florida?
Screwworm.
Yeah.
Yeah, but not for the Texas-Mexico border, Alan. That still comes back every year.
Really?
Yeah, because it's not geographically isolated like Florida is.
I thought they had finally wiped it out from Mexico as well.
I haven't heard that one. I would love to know that.
Yeah.
You
Yeah,
have
my
more-
grandfather was involved in that original.
Is that right?
Yeah.
It was we'll get into that some other time, but it's a remarkable story.
Yeah, that's probably for TWiP.
Exact, or for TWiF. The F is for flies.
Flies. I don't know if we'll ever get to that one. It says here it's believed that wildlife populations cannot maintain the Rinderpest virus indefinitely. So far, there haven't been any outbreaks in a couple of years anywhere. I think it was eradicated in Europe a number of years ago.
They say the last epidemic in Africa was 2001, so that's a long time. They must have a bunch of epidemiologists watching
That's
this
right.
and
They
deciding,
do.
Yeah.
They
deciding when they can actually make the declaration.
Right.
So
be a
there are
miracle if
three.
that
There's
happened.
one serotype, so you make one vaccine and you can get rid of it. And
What does that tell you about evolution in this
Well,
case?
it's like measles is the same. There's one serotype, and we use one vaccine to immunize everyone. And they say there are 3 genetic lineages, and 2 of the 3 have been gone. Haven't seen them in years. The disappearance of Rinderpest would be the first worldwide eradication of an animal pathogen.
Wow.
I don't know. Aren't we animals?
We are,
Yeah.
actually.
And of course, smallpox is the other one.
Yep.
So I don't know. I mean, I think it's iffy to say it's not here because you can't sample everywhere.
No.
As someone once told me who's involved in the polio eradication, saying a virus isn't present is only as good as your sampling, and
This
you
is
can't
so true.
sample everything,
No,
right?
that's right.
So I was always worried about this being an animal virus. It could be out there. Who knows? But they're immunizing, they're checking, and they haven't seen anything in a long time. So maybe it's true.
It
We'll
would
see.
be great if that happened. It would be great.
Well, I think it's already improved the situation because there hasn't been an outbreak in a long time, right?
That's right. I would agree.
So that would be good.
Now there is still the issue that now comes up with smallpox, and I understand
Laboratory.
Rinderpest was investigated for this, at least in the U.S., which means
Sure.
it was probably studied in Russia as well,
Yeah.
as a biological weapon.
Yes, and I'm
Now,
sure
post-eradication, if you stop vaccinating, that would make anybody in possession of Rinderpest
Yeah.
potentially very powerful.
that's right.
Is that the plan to stop immunizing at some point here?
Well, they say FAO says they're shutting down their field operations,
Interesting.
which I would expect for Rinderpest. They're not shutting down all their field operations.
right. Is that a live virus vaccine, by the way?
Yes,
Oh, yes, it is.
it's a live attenuated vaccine. It was
Goodness.
developed in the classical fashion by a guy named Walter Plowright. This one I paid a little bit of attention to. So
Nice.
it was something like 90 passages in
Yeah, yeah.
cell culture.
Yep.
And they've looked at the vaccine and figured out that during that period of time, it accumulated a bunch of different lesions in a bunch of different genes. And they've actually done the experiments to sort out individual lesions, look at the contribution of individual mutations to the overall attenuation of the virus, and there's no one culprit. It's a constellation of all of the different effects.
Sounds
But
great.
what that means is that we have full published sequence, and we know which mutations are in it that would have to be
Yeah.
back reverted.
Yeah.
Just
And
saying.
this gentleman, Walter Plowright, who developed the vaccine, just died in February.
Oh,
I
gee.
dug up a bunch of obituaries to him. So he's a big deal in the Rinderpest world. Saved a lot of people.
Too bad he didn't live long enough to see it completely disappear.
Yeah,
I've never heard of them.
you have now.
I
Well,
Well,
mean,
that's
listen to
the
TWiV.
problem with agricultural
Yeah,
research. It
exactly.
really it gets no airtime.
The problem also is that it's an African-based disease, although it's not only in Africa, but it's Afrocentric.
they got rid of it in Europe.
Yeah,
That was
but
a
that was a
problem.
while back.
But that's the point that Alan made is that, we don't learn much about agricultural pathogens. It's too bad because we should know this.
Right. Most people in the modern world think that meat comes from a styrofoam package.
And it does.
It does, yes, but there's a story behind that.
Indeed.
And by the way, measles is thought to have evolved from this virus. We've talked about this before. Rinderpest, and when cattle were first farmed by humans, husbanded,
Yeah.
15,000 years ago,
Oh, earlier than
Domesticated?
that. Probably earlier than that.
earlier.
Yeah.
That's when this Rinderpest began to transmit very effectively, and then we're farming them and right next to them, so it jumped to us and became measles. It's very close. It's the closest of any of these related viruses. So,
do cattle get SSPE then?
They don't live long enough.
well, the Rinderpest can be
Get dead,
100% fatal. Yeah. So,
Yeah.
you see, measles is not. So that's why. So,
So yeah, I found one paper. There's, I think, a bunch of papers looking at the possible evolution of measles from Rinderpest, but I found one that using phylogenetic methods suggests that the measles came about from Rinderpest between the 11th and 12th centuries.
I'll be darned.
So I guess the way this is done is just with bioinformatics, right?
yeah, sure.
You look at the relationship between the two and try and figure out how long ago they diverged, which, as we will see, is not necessarily a precise method. But it's interesting because we can say that with fair confidence that HIV evolved from SIV because in our sort of living consciousness, we can point to a time when humans didn't have HIV.
Right.
But you can't, I don't think that their records or histories are good enough to go back and say when and where and if measles just suddenly arose in the human population or if it's been around for a while.
Yeah.
So this is, I guess, all bioinformatics stuff.
Yeah, it's all bioinformatics. You compare the sequences, you make phylogenetic trees, and you estimate when they diverged.
So, Vince, here's a general question for the audience of three. How many viral infections of humans are strictly restricted to humans?
Ooh. I don't know if
Hey,
know.
come on, guys. I'm the
Well,
foil here.
There's a bunch.
We got measles and polio.
Yeah.
The herpes virus infections that are human herpes viruses they don't cross species very well. Of the herpes viruses, there's only one that I know of that can infect humans as a zoonosis, and that's herpes samurai. And that's not a natural situation. That's mostly people working in laboratories that...
what about the rhino viruses?
No, rhinos don't seem to infect animals.
Right, except other rhinos.
Smallpox. Did I say the papilloma viruses?
No.
You did not, but you should.
So I think they're quite a few.
The polyoma viruses are very species specific. So there's a lot. In fact, I think interspecies transmission is a much rarer situation than viruses being species specific. You have families that infect a wide variety of species, but within a given species, a given genus or whatever species of virus will stick with that species of animal.
Right. So the other question then I have is that if we were to go back through the fossil record of just hominids and take the small deposit of DNA in the bone of the brain case, which is what they use to do the sequencing from, could you find remnants of this virus back until it wasn't there? Has anyone ever done that?
I don't know if they've tried, but no one's reported finding anything.
Because, I mean, this sounds like a wonderful example of here's an animal disease that jumped into humans, and we can almost pinpoint when that happened.
The only time we see it is in the genome, endogenous viruses.
Right, so when did we get our others that are now just restricted to us? And
I mean, the problem with the RNA viruses is that they're labile and they would go away unless the specimens are well preserved, like the ones in Alaska in the permafrost from which they got the 1918 influenza virus.
Or the Iceman that they had covered from Switzerland.
And none of those are even long enough. I mean, you're talking about maybe 10 or 12,000 years.
Yeah, that's not long enough.
We need to go back hundreds of thousands, millions of years.
Here.
My guess is that a lot of these things have been evolving with humans for a very, very long time.
Right. And has anybody ever isolated a viral sequence from amber?
Not that I know of. You got any sequence from amber?
That was a general question, gentlemen.
That's a movie. That's all I know. And I don't know of any sequence from amber. I could be wrong. But
there's a lot of sequences.
Do you know, Alan?
There are certainly critter sequences from amber, but I don't know of any viral sequences that have come out of that.
Okay. I didn't mean to divert us away from the topic so much, but
yeah, that's okay.
That's why you're here.
Time.
That's why we hire you.
Right.
I'm a diversionary tactic.
So you're previewing one of our other subjects.
Well, Vince has prepped me on this one. He's prepped me on this one.
Okay, well, we'll see if it's eradicated.
We're going to be around next year, Vince.
Yeah, we will be. So
we're going to be able to say whether this actually happened or not.
And of course, if there's an outbreak, which would be a big deal, it would set back.
It'd be a huge deal.
Because this has cost a lot of
money. But it could also come from a laboratory. Do you think Rinderpest could come out of a laboratory like Plum Island?
Actually, it will be very interesting. I wonder what sort of measures they're taking to make sure that it's not going to get out of laboratories. Of course, when they eradicated smallpox, they went through the effort to collect all the virus out of laboratories and make sure that, to the best of their knowledge, it only exists in 2 places now under lock and key. And I guess they're at least cataloging polio in preparation for doing something similar. Right. I would assume they've done something similar for Rinderpest.
So is it going to be a highly contained virus at some point? Maybe it already is.
I don't know. Maybe so.
Let's see. Let's just try and find it right now. Rinderpest. I can't see the screen, Dickson.
I can.
According to, let's see, bringing up a story from the Guardian paper in the UK.
Ooh, look. Manual on the preparation of Rinderpest contingency plans. So I guess they are going to restrict access to it. I wonder if they're going to destroy all stocks. I have not seen any of that.
No one's ever done
so
that.
stocks. According to the Guardian story that I'm looking at now, stocks of Rinderpest virus will be kept in high security laboratories, including the Institute for Animal Health Purbright Laboratory in Surrey, UK, which appears to be the researchers they interviewed for this piece. So I guess the plan is to keep the virus, at least for a little while, and I guess they'll have the same debate that we have now about smallpox.
Of course they will.
Should we wipe out those stocks? Do we know that those are all the stocks? And as I said, if the U.S. worked on it during the Cold War as a weapon, as we know we did, then I would bet money that Russia worked on it as well. So
sure.
Who knows where those stocks are?
I just found a terrific book on the whole eradication issue put out by the FAO. Put a link to it here. Manual on the preparation of Rinderpest contingency plans. Multiple chapters. Very good.
That's a hell of an organization, by the way. I have a deep respect for
FAO?
it. Yeah, terrific.
Yeah, they do an enormous amount with very little.
Yeah, they sell toys down here on Fifth Avenue.
That's Schwartz.
Sorry.
Vince, that's a different FAO.
Okay, moving on. Moving on to our next story, which is endogenous hepatinoviruses in birds. Hepatinoviruses. Hepatitis B virus, TWiV 80. Michael Bouchard, we talked about them. I don't think you were here, Dickson.
I was not. Not for that particular
That was in your
episode.
period of absence.
It was my winter of discontent. No, that's not true.
It's from the Department of Biology in University of Texas, Arlington, which is where the World Series is now.
It is true.
Right?
It is
Arlington,
true.
Texas. Where is Arlington?
It's next to Dallas-Fort Worth. It's
see.
sort of directly in between both of those places.
I see. By Clément Gilbert and Cédric Féchot.
Couple of nice Texan names.
Yeah.
There you go. That's right.
Came from France to where the money is.
Yeah.
And in this study, very interesting. Hepatinoviruses, hepatitis B virus, are DNA viruses enveloped with a reverse transcriptase, part of their life cycle. Very unusual. They do not integrate into the host genome, though, unlike retroviruses. It's an essential part of their life cycle. And what they did here was simply to take the sequence of duck hepatitis B virus, their duck and squirrel and human versions, and search the sequence database and said, is there anything there besides other hepatinoviruses? Okay. They were looking for endogenous hepatinoviruses. I don't know. I'm not sure why. I guess since we've now had reports of endogenous.
Because we can.
Yeah.
Right. This is a virus that you would expect would be able to endogenize.
I guess because it has, well, not because of the reverse transcriptase, but because it's in the nucleus. So
right, it's in the nucleus. It's
that.
got DNA. It's
and they make a nice.
We've seen a number. We've seen a number of these recently where people are. I think this is mostly it's genetic genetic fossils. People are looking for
Sure.
basically genomic fossil evidence of the existence of viruses way back in time. So we had a TWiV some time ago with, what was it, Bornavirus?
Bornaviruses, yeah.
And some others. And so this is just another example of looking for a fossil record of viruses by looking at genomic sequences.
You know, the interesting thing is that, so we had Bornavirus, retroviruses, of course, Bornaviruses,
and filoviruses, that's it.
They don't mention any of those here, so I have a feeling this was submitted before that came
no,
out.
I thought I saw maybe it's in something else that I read.
Because they do mention
some reference to those other publications.
They mentioned some other viruses that I didn't know endogenized. For example, Gemini-like viruses in tobacco, endogenous viruses in tobacco, and non-retroviral RNA viruses like totovirus-like and M2 killer-like viruses in fungi, and flaviviruses in mosquito. I didn't know that flavies were known to be endogenous in mosquitoes.
It's not an unexpected consequence of total association, though, because most flaviviruses are transmitted by mosquitoes. Yeah. So there you go.
So, I mean, the thing is that up to a year ago, I wouldn't have expected a non-retrovirus to be endogenized, but now we know that it can happen. And now we have paleovirology is what they call this field for many more viruses other than retroviruses. So,
and a major reason this field is exploding like this is, looking through this paper, I don't think these guys had to pick up a pipette.
They didn't.
No, it's all computer stuff.
It's all in the computer.
Wait a minute, that's not true. They PCR'd. They PCR'd some sequences out to confirm what they have. Nevertheless, right.
It's most of the work. I mean, you don't have the whole period of trying to figure out if you're even going to find anything. They do most of the preliminary work. Just searching the databases because all this information is out there, and most of this information comes from these whole genome sequencing projects where they have so much data they just dump it into the database and let other people analyze it. So that's a lot of what's going on here is just mining the data that we already have from all these genome sequencing efforts.
So they do have a sentence here in the introduction. Likewise, the recent discovery of multiple endogenous Bornaviruses and filoviruses and diverse blah blah blah.
All right, so it was there. Got it. So basically what they do is they search the entire GenBank sequence database, which is big, with duck hepatitis proteins. And they got 15 hepatinovirus-like hits in the zebra finch genome.
Wow.
That's it. Just the zebra finch.
Which is really weird. Why the zebra finch?
You know anything about that, Dickson, zebra finches?
It's an experimental animal. It's used in the laboratory all the time.
Is it all over the world?
Well, that bird is because people work on it for other reasons.
They brought it around, yeah.
Yeah, but it's indigenous to like Australia and Indonesia, I guess.
Do you remember what the original designation for hepatitis virus was? Australia antigen.
Yeah.
Ah, that's right.
Yeah, that's a coincidence.
Yeah, not just a coincidence.
With zebra finches, it must be a coincidence.
Well,
he's a cute little bird, I'll tell you.
True, they are.
Yeah, they're nice birds. So they have 15 different, their integrations into 10 different chromosomes, and the identity 55 to 75% nucleotide similarity to the duck hepatitis B genome, which is pretty close considering.
Right. So the reason you come up with so many zebrafish hits in the database is you searched with a bird. Zebra finch. Yes.
There is a zebrafish too.
You searched with a bird virus.
Yeah.
And you're going to get bird hits out of the database, I would assume. And what bird is best represented in the databases?
Zebrafinch. Yeah.
Exactly.
So the total genome is done.
So I don't think we've sequenced owls and
quail is there too, and chicken. They just did the chicken genome.
They just did the chicken.
The whole Science magazine was devoted to it.
Right.
So they have a nice map in this paper, and you can see that they have a genome map on the top, and then where all these pieces came from. So they're pieces from the left hand of the genome and the right. Well, this is a circular genome, but they've made it linear.
So I want to interject something here because the hepatitis viruses, you may have said, do not integrate as an obligatory part of their life cycle.
Right.
But they do have they do make double-stranded DNA
Right.
as part of their replication cycle, and that double-stranded DNA spends time in the nucleus. And anytime you stick a DNA molecule into the nucleus, there's a certain probability that it's going to get integrated.
That's right.
Anybody who's worked in cell culture and done transfections knows that. If you transfect a plasmid into cells, it just gets integrated higgledy-piggledy, non-homologous recombination anywhere it wants to go. And most of the places are of no consequence, so it's not a problem. So even though this thing does not, to anthropomorphize it, deliberately integrate into the genome, there's a certain probability that because the DNA spends time there, that it will become endogenized. And so you don't necessarily expect to see full-length genomes or anything like that. You're going to get little pieces that show up here and there, and they could be any part and they can be anywhere. And that's what's represented here. There's little fragments of the genome scattered all over the chromosomes of this finch, little pieces.
And these are all dead. They don't ever give rise to virus.
Right.
Now, in contrast to the retroviruses, where when you get the full genome in, it can give rise to more viruses.
Right.
These are just pieces. These are probably of no consequence to the virus, but for us, they're experimentally useful for timing, right? What I found cool was that one of these pieces was 97% identical, and they said it must have duplicated at some point in the genome. So that gives you a clock. You can say, how different are they? So how old might they be? So
Not only are they 90% identical, but they're right next to each other on the same chromosome.
Right.
So that supports the notion that it was a duplication.
So they used that to do a preliminary calculation of how old this is, and they came up with like 9 million years.
Right, because we have reasonably good clocks, or we think we have reasonably good clocks for mutation rates in vertebrates.
Right. So you can see how different are these two duplications, so you get a rough idea of how long ago they duplicated. So 9 million years ago. So that means these were there at least 9 million years ago. But the other way that's been done with retros is to look at species, phylogenetically related species, and say, where is the point going back from the finch that we can see where the species separated, and do we still find these sequences? So that's what they did. This is where they picked up a pipette. They did some PCR and ancestral birds to the finch, and they find it, and they can then say, here's the point where it bifurcated, and it wasn't present in the ancestor, this duplicated insert. And they then come up with this estimate that this virus, these pieces of these hepatitis virus genomes, been in there for 19 to 40 million years. It's a big range.
Yeah,
I'd say.
but still, it's a long time.
It's a long time.
Yep.
It's way back there.
Which is as long as any other estimate I've seen, even for the retrovirus. I think the herpes viruses are estimated to be 140 million years old.
Wow.
Based on just clocks, which would put them in there with the dinosaurs.
Absolutely.
It would be a pretty big fever sore, wouldn't it?
I'd say.
But this is old, 19 to 40 million years. So that's pretty neat that you can say that. And
so what surprised them was that apparently if you look at the existing hepatinoviruses, you get a certain mutation rate, a certain apparent mutation rate that is 1,000 times faster than the apparent mutation rate that you get by looking at the drift in these, the difference between the current sequences and these sequences from these birds. So there's a discrepancy there between the apparent mutation rate that's going on now and the accumulated mutations between the present and this 19 million years ago. And there's a lot of speculation as to why that difference might be.
I'm getting a lot of jitter on my end of the line. Are other people hearing okay?
You sound good.
Fine.
Everything sounds good to me.
You could unplug you can unplug and plug in again, or you have an analog thing there, or you have a USB converter.
I have a USB. I'll unplug and replug.
Go ahead.
Alan will be back in a moment. After these words from...
Yeah, we don't know the reason for this discrepancy in the mutation rate. I mean, one thing they say is that maybe these viruses at one time were always in the genome, and only recently they begin to replicate as exogenous viruses.
I'm not buying that.
I don't buy that.
Right.
I don't think they changed that dramatically.
Go to Plan B.
Yeah, there is no Plan B.
But to me, a theme that comes up that we were talking about earlier is that because in particular with RNA viruses or viruses that don't have proofreading enzymes that are their replicases, and that includes these because they use a reverse transcriptase and that doesn't have a proofreading function, the mutation rate that you see by looking at the viruses that are extant gives you, if you look at that, it suggests a much faster evolution than you get by looking at the fossil record. There's a disconnect here somehow. As if one of the things they suggest in this, for example, is that maybe these viruses really do evolve fast, but occasionally they go through some sort of genetic bottleneck that limits the actual sequences that get passed on in the long run. Okay, so to me, one of the messages is that there are a lot of problems in trying to do these phylogenetic trees of viruses and really dating their origins, and things we're going to have to figure out.
Couldn't it simply be, Rich, that these pieces went in 20 million years ago and they've been evolving at the rate of the genome?
Oh yeah, I think that's true. But the point is that if you compare those sequences with the viruses that are currently here, and if you look at the apparent mutation rates among the viruses that are currently here, you would have expected much more divergence between the stuff they're finding in the genome and the current stuff.
Okay.
Right? Yeah. Like 1,000 times more.
Interesting.
So there's a disconnect there between this fossil record and how much drift there's been that was from the virus, if we look at it 19 million years ago, and the apparent mutation rate of these viruses currently. There's a big disconnect there.
Well, except that the viruses are probably going through punctuated equilibrium at each step where they jump to a species and they're going to relatively stabilize. And yeah, they could have a faster mutation rate if you pick out individual plaques and sequence them, but aren't they relatively stable over time?
This is an element of the whole quasi-species thing that I don't really understand fully, okay? And that I think goes back to what I was talking about before, the idea of bottlenecks. There's even though you have a lot of mutation, in the long run, there's only a few of these that make it in the long run.
Right, they average toward a particular consensus because that's what works best for infecting this particular host.
You can have also major shifts, depending on the selections or the bottlenecks, as you say. That
we ought to get somebody on sometime to try and explain quasi-species.
That would be good.
We'd have to resurrect Stephen Jay Gould for this punctuated evolutionary.
Is he dead?
I'm afraid so.
Yes, he is. Yeah.
The last thing we should mention here is that we don't actually know if there are any circulating zebra finch hepatinoviruses today. No one has ever described them. They're in other birds, but as they say, we should probably look.
I bet some people are looking for those now.
Yeah.
It would be hard not to look after this.
Yeah, I mean, it's easy now. You go out and collect some and look in them.
Sure.
And keep collecting until you find them.
Well, and check your lab stocks, too.
Lab stocks of zebra finches?
Sure.
Sure, I got a whole lab full of zebra finches.
Good. There you go. You can set up some PCRs tonight. Anyway, that's pretty cool. Paleovirology. And
I think we're going to see a lot more of that.
You bet.
Yeah, it's really interesting stuff.
The last story is also dealing with endogenous viruses.
Fins.
These are retroviruses.
Uh-oh.
The title is Identification of a Receptor for an Extinct Virus.
An extinct?
I'm sorry, I messed it up. Extinct.
It reads extinct. I can read that from here.
It's PNAS. I thought that was a neat title.
Yeah.
From Paul B. Nash. Steve Saul, Stuart Neal, and Paul B. Nash here in New York City, the Aaron Diamond AIDS Research Center. So they looked at two endogenous viruses of chimpanzees. They're called chimpanzee endogenous retrovirus 1 and 2. And these are in the chimp genome, but they don't make viruses. They're defective. Okay? And they're not in the human genome. We don't have them. So at some point when humans and chimps split, I guess we didn't have them.
We're closely related to gorillas than we are to chimps.
Yeah, but we did have an common ancestor, right?
Well, yes, but I think probably you'd have to go to gorillas and humans and then to another stem cell before you got to chimps.
So they say here the viruses are of particular interest because they're absent in the human genome, although they're in the chimp, bonobo, gorilla, and Old World monkeys. Thus, they both apparently replicated after divergence of the human and chimp lineages 6 million years ago, with zoonoses resulting in endogenization in diverse Old World primates, but not in people.
And being retroviruses, these do integrate as an obligatory part of their life cycle.
Yeah.
So they're much more likely to leave this sort of fossil record.
Yeah.
So if humans had ever had them, you would expect to see them in the human genome.
Right.
And they say here, which is interesting, the viruses are assumed to be extinct. We don't find the viruses floating around, or what we call exogenous viruses. Although without exhaustive sampling, it is nearly impossible to definitively demonstrate that intact exogenous relatives are not currently replicated in some modern primate species. So I think that's important.
Right.
You can never say a virus is extinct or eradicated, right?
That's true.
It's very hard to prove a negative.
You know, the other thing that's really remarkable here is that new primate species are being discovered every year, which is hard to believe because you can see these things, right? The last couple that were discovered were tiny little primates.
Where?
I think one of them was in Madagascar, and the other one was, I think, somewhere in Papua New Guinea.
Yeah, you must have to dig around in the wild to find a new.
And they were nocturnal species that hardly ever come out in the daytime, so they're hard to see. But the fact is that we're still discovering hosts. So the viruses will obviously come forth once the hosts have been identified. That's my view.
So what they did in this paper is basically to take the envelope glycoprotein of these endogenous viruses and reconstruct them so that they worked properly. Because they have all kinds of mutations in the genome, but they get sequences from different species and assemble something that looks correct. And then they take that envelope and express it on another retrovirus. They put it on a murine leukemia virus. Almost sneezed, but not quite. It'll happen in a minute. He took his headset off.
I had to yawn. I'm sorry.
Oh, that was yawning. Sorry.
I didn't want to break the rhythm of Vince's cadence.
So they reconstructed the envelope and made what we call pseudotypes, which is just the envelope of one retrovirus on a different retrovirus. Oh. So it's this chimp endogenous envelope on a murine virus. And then you can use that to infect cells and do experiments. You don't have to have the whole chimp virus reconstructed. And they found that human cells were perfectly susceptible to this virus. And they only...
Right. So that doesn't explain why. That doesn't explain why the chimp virus didn't infect humans.
Yeah.
Exactly.
So we don't have it in our genome, and so that's not because we weren't infectable, because we apparently are. And they found that hamster cells were not permissive. They don't have the receptor. So they used that to clone the receptor.
What is the receptor, Vince?
It's a copper transporter molecule.
No,
it's one of these
like ceruloplasm?
I don't know what you're talking about.
That's a copper receptor.
There's a transmembrane protein.
No, this is the protein in your blood that transmits
that binds
copper. Yeah,
do we need copper?
Copper transport protein 1.
Do we need copper? Of course we do.
For our pipes.
That's it.
You know, when you take apart a house, the plumbers come and take all the old copper pipes.
It's very valuable stuff. Well, we have zinc fingers and probably copper palms.
Well, in a lot of places, also electrical wiring gets stolen, which is a little obscure.
Yeah, that's right.
So this is a multi-transmembrane protein that moves copper across the membrane. And so this virus is evolved to bind to this and use it to get into cells. And the hamster version has mutations in the N-terminus that make it not work for a virus receptor.
Neat.
So that's the reason why they can't be infected. But,
or rather, the primate lineage version has mutations that make it work as a virus receptor.
Which came first?
Is right.
So that is interesting. It's cool. It's a receptor for this virus. Sure. And the neat thing is why it didn't, as Rich said before, why don't we have it? And we don't know why we don't have this virus. Maybe we weren't in the right place to get it. Maybe it left us. Maybe we did have it for a while and it left us.
We haven't fought it off.
But the whole point here is that you can use these endogenous viruses to start looking at these questions. They say here, what are the problems with getting ancient virus infections? Now we can sort of look at them. But what I thought about, I don't know if you guys remember, we talked about a paper from this group some time ago where they reconstructed one of the human endogenous retroviruses. Guys remember that?
Yeah, I was going to ask about more detail on that. I don't think I was on that show. You did that on TWiV, right?
Mm-hmm. Maybe not.
I don't think I was on that show, but I remember the fact that they did it. What virus was that?
It's called HERV-K. It's the closest to a functional human endogenous retrovirus, because all our endogenous retroviruses are dead. But the HERV-K is...
Or non-functional.
Non-functional, sorry. The non-functional. And HERV-K is probably the most recent one to have come into our genome. So they just corrected it. They couldn't get the envelope to work, but they got everything else. They got the GAG protein to work in the pol. So they reconstructed that virus and did some work with it. But I was just thinking it would be interesting to get the receptor for that in human cells, right? But they don't have the functional envelope, so you can't do that until you have that, and they can't do it.
Well, one of the interesting things about this paper to me was just the technology of actually identifying the receptor.
Yep.
They took, they made a library and a retrovirus, transfected that into a cell that they had discovered in their screen that was non-permissive and could identify that it was non-permissive because it didn't have the receptors. So
that's the hamster cell, yeah.
Right. So you transfect this retrovirus library into the non-permissive cell and then screen for cells that are positive. And then, because it's integrated along with a retrovirus, you can PCR out the gene the candidate gene that's been transfected because you know what it's anchored to, the retrovirus. You know a thing or two about cloning receptors, Vincent.
Yes, this is very close to home, and that's a little more advanced technology than we had.
I mean, you had a much rougher go than that, didn't you?
You know, in fact, a few weeks ago, I was asked to write a commentary for this paper for when it was published, and that's why probably, because we did this years ago. Unfortunately, I wasn't able to do it. But so we had also, when we identified the polio receptor, we had mouse cells which lack the polio receptor. We put human DNA into those cells and looked for mouse cells that could now be infected with polio.
But the transfected DNA probably wasn't tagged, is that right?
Not by us, but it's naturally tagged by a repetitive sequence called an ALU element, a highly repetitive human sequence element present in about 1 every 5 kb of DNA. So we could use that.
Okay, so you could identify the human DNA over the mouse DNA background using it. Okay, fine.
I mean, the real problem was when you can't infect the mouse cells with polio because it will kill them and you won't have them anymore. So we actually were fortunate to get from a collaborator, an antibody that blocked polio infection, which we assumed was directed against the receptor. So we screened the mouse transformants with that and found some that expressed the receptor.
All right.
And then we used the ALU to pull it out. So it's basically the same idea, except they have a little more advanced technology. It's a bit easier. Yeah.
Okay.
Yeah, the technology here is very cool.
Yeah.
It's a neat paper. So that's why I picked it. And they end up by saying this work demonstrates that paleovirology has the potential to both identify novel host factors usurped by ancient viruses, as well as indicate the limitations that hosts may have faced in avoiding such exploitation. Neat.
Great.
Yeah, I like this paper. It's good.
Indeed.
That brings us to...
Sounds like letters. Vince.
Letters to the editors.
Letters to the editors.
We are men of letters.
We are. A, B, C.
The first is from Timothy, who is a postdoc here in New York at the Rockefeller University in the lab of Charles Rice.
It's a good place.
On hepatitis C virus. Dear Vince and Alan.
Forget about it, Rich. We're out of it.
I don't know why. I must have been...
I can handle it.
Yeah, me too.
It's because it's a show Alan and I did on XMRV, yeah.
All right.
Being a seasoned virologist and a science writer, I was hoping you could discuss the interface between basic science, public health, the media, and the general public.
How much time do you have?
Yeah.
That's what Vince just did. He looked at his watch and said, I don't know.
This is an hour show. Of time doing this, and it's an important topic.
Tell him to call me.
Well, he wants this discussed in front of everyone.
Yeah,
well,
all
right.
We'll
Alan, go for it.
I'm sure we all have ideas. Let me finish his email. That was only the first
That
sentence.
This is an 8-part question.
While I was listening to you discuss the controversy surrounding the potential involvement of XMRV in CFS, chronic fatigue syndrome, it unfortunately reminded me of the Andrew Wakefield story. Like the Wakefield saga, it seems as though the Whitmore-Peterson study has given a glimmer of hope to a patient population suffering from a difficult-to-diagnose disease, as we saw with the Wakefield study. Even though several studies disproved the association between the MMR vaccine and autism, many people were reluctant to believe it. Alas, pipettes are no match for hope.
That's a great line. I like that.
Is there any way to temper the media's response to studies like these in order to send a message that's more realistic? Relatedly, the media response to H1N1 pandemic threat was especially painful to watch. The media could be one of the most effective arms of the public health machine, but oftentimes, instead of helping, it creates a world of hurt. What is your take on this situation? How can the scientific community disseminate more responsible and accurate information to the public? Is this even possible since the media's business revolves around attention grabbing? Are pipettes no match for the dollar? Keep up the good work until next week. All right.
Go for it, Alan.
Go for it, Alan.
So I guess I'll take this in order. The XMRV CFS and drawing a parallel between that and the Wakefield story, I think it's premature to say that. I mean, in the autism vaccine case, you had one poorly done study that was subsequently disproven by just legions of enormous studies, one after another, because this would have been a very important finding if true. There was no evidence that it was true based on the original study, but it was clearly something that was important to restore public confidence in. And I don't think there was ever any serious contention for that theory. In the case of XMRV and CFS, what you have is a preliminary paper that was not shoddy. I mean, it was, from what I could tell of the data, it looked like a pretty good paper. And then subsequently, there have been some papers of varying quality that have not found a link, and there's been at least one that I think was another very good paper that found murine-like retroviruses associated with CFS. Notice I didn't say XMRV associated with CFS, but the Alter paper, which we talked about extensively, it showed that maybe there is something going on here. So with XMRV, or murine-like retroviruses, I should say, and CFS, I would say the jury's still out. Now, I do know that there's a somewhat wider story here, which is the WPI, and particularly Mikovits, the main author on the first XMRV CFS paper, have subsequently kind of gone a little off the rails. I understand that she's now claiming XMRV causes autism, and she's way out ahead of the published data, at least, which is disturbing, and I think does undercut her credibility. But we have an independent study that says something pretty similar. So I don't think I would tar this with that same brush. I think this may turn out to have been a false lead, but I don't think that.
Well, Alan, don't you think the investigator has the responsibility then of qualifying the results and saying that more results are due to come out, and you should hold off on sensationalizing the story?
My criticism of the WPI work is that they were very, very promotional of one particular interpretation of the data, as if it were a foregone conclusion that this was absolutely all nailed down, when in fact we all know that one study does not do that. So yes, they should have been a little more circumspect than they were, but I don't think that puts it into the same caliber as what Wakefield did with a poorly structured and apparently unethical study, which came to a completely wacky conclusion. So I wouldn't quite put those in that same category.
I mean, most of the reporters, no matter how much science background they've had, with you as an exception, are unable to judge the real science of this, and they have to depend upon the knowledge of the investigator to tell them. And so a lot of these people are just being told things that the investigator, I'm not saying, well, everybody does this, but I know that this has happened. This increases your chances for getting refunded if this thing hits the newsstands and everybody's aware of it. And then all of a sudden there's a study section that meets, and the next thing you know, you got your grant. And that's, I saw this happen myself while I served on a study section down in Washington. And I was disturbed by it because I I mean, some of those things turned out to be true and some didn't. And I understand where this guy's coming from at Rockefeller. I mean, I think, it's great enough when you make a small discovery that turns out to be true.
Yes.
You cling to those wonderful discoveries of nature that you were the first one to see without having to claim that I can cure every other disease by simply finding a retrovirus in somebody's genome.
Right.
I think that's what this guy's trying to say.
Well, the key here is there any way to temper the media's response?
Well, it's up to the investigator to temper the response.
I don't think in free countries that you can do that, and I wouldn't want to live in a place where you could.
Oh,
that's what David Baltimore said. In a free economy, this is always going to happen, basically.
Yeah.
So when I think about this, one of the things I think about is the way political polls are reported. Where they say blah 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 blah, and then they say these data are accurate within a certain percent. So people at least are used to seeing in the standard reporting of polls some sort of qualifier,
right?
That at least alerts them to the fact, to the idea that this may not be true, or there's some uncertainty here, a certain measured uncertainty.
That's right.
It would be nice if we could routinely apply some sort of cautionary boundary to the science reporting. I don't know how to do it, but
and in fact, reporters are well versed in reporters who cover politics often have no math background at all, but they're well versed in reading a poll, and if a contest is within the margin of error, they'll refer to it as a dead heat.
Yeah.
So and so was ahead by 2 points within the margin of error of the poll.
Right.
So that's a very common construct, and that sort of thing can certainly get out there and become common practice. I think the problem here is that the complexities are not things that you can reduce to a specific percentage.
Right.
It's not a baseball statistic. It's not a political poll. It's not something where you can say, XMRV could cause chronic fatigue syndrome within a margin of error of X percent.
That's right. The other nice thing that the best science reporters do, and that is that they check with other scientists. And they have them quoted in the articles. And what do you think of this? And how good do you think that data is? Using their expertise to sort of qualify the original
yes, that should be. It should be standard practice, and in professional journalism, it is. I think what we see going on now is there are a lot of news outlets online that are referred to as news outlets, but in fact, all they're really doing is spidering press release pages and reprinting them.
Yeah.
So we come back to the same thing that we've had before, same conclusion we've drawn before in this discussion, and that is, you can't really properly temper the media ultimately, and it's the public's.
Here.
The public's response. We have to have a scientifically educated public.
And to some extent, it is also the scientist's responsibility.
Yes.
Why do you, gentlemen?
I mean, you can't get these press releases reprinted if somebody didn't write them.
Why do you think we're doing this show?
Yes.
That's the whole purpose of this week in virology and this week in parasitism is to temper the scientific literature and to give opinions that might have some relevance to the future.
Yeah, but we're preaching to a very small fraction of...
Here's one guy who listens.
Yeah,
that's my answer.
I mean, it's not... we keep doing it. Hopefully someday we reach 1 million people.
Well,
but
that's the point.
The fact is that in the first science paper, they said at the end, this does not prove that XMRV causes chronic fatigue.
The press missed that little statement.
More studies are needed, but people miss it, not the press. I mean, they don't probably include it in their articles.
And there's a technical issue here too, because you have to say things like that in a paper for it to pass peer review. Mm-hmm. And so, yes, in the paper, they say there are all these caveats because that's mandatory. And in science, we actually do enforce. You have to spell out what's wrong with your own work. Or somebody else will do it for you. But press releases are not peer-reviewed.
No.
And unfortunately, the press release is always going to get wider circulation as long as the publication is closed and as long as there is a running habit in the press of not linking directly to the primary paper.
And finally, of the reporter not sending it back to the investigator for checking.
If the investigator would have checked it. I think that's where you need outside opinions.
Yeah.
Right.
The problem is the nature of science is such that people will never take away comfort from it. As Alan said before, you can never prove a negative. you can never prove without doubt that thimerosal doesn't do something. You can only prove within a certain limit, right? And most people do not get it. So if you say we've done 12 studies which show no statistical association of autism with having thimerosal in vaccines, but the numbers are only good to a level of 1 in a million, say, the public will say, well, then you don't really know, do you? So I'm going back to my voodoo recipes.
Right, you're talking to an audience that supports state lotteries around the country.
Here. Yeah, no, it's a propensity of people to want to hear what they believe already. And, if you're one of these people suffering from chronic fatigue syndrome, and they're accusing you of just being lazy on the job, and all of a sudden there's this virus that causes it, or may cause it, now you have an excuse to go back to them and see, it wasn't me after all, it was my infection that did it.
I think that having a cause for anything, like autism, where you don't know, that's a big deal, sure.
That's right. Well, autism might be a little different, but I think these people that are suffering from chronic fatigue syndrome are still maintaining a semi-normal life. They're trying to go to work, they're trying to behave, but they can't because they're constantly tired, obviously. Sure. So here's, it's not an excuse-seeking behavior, but nonetheless they cling to these little bits and pieces of evidence because they're desperate for a solution.
Pipettes are no match for hope.
Here.
Exactly.
Or money. Money.
In fact, I don't know, Timothy, if you haven't read Autism's False Prophets by Paul Offit, it's a previous pick on TWiV. He goes through all of these issues, and he says in the end, science is no match for a mother's intuition. The mother said, I saw this happen to my child after getting the vaccine. I know it was caused by the vaccine. There's nothing you can do to combat that. Right. And, polls have shown that Americans are incredibly influenced by faith and black magic and all sorts of non-scientific, maybe others, but in the book he talks about polls and taken here, all non-scientific things. Yeah. So,
you can't reason somebody out of a position they didn't reason themselves into.
Well, that's right.
So we do our job, but I think it require many generations of people better educated in science.
Well, one of the biggest,
I don't know if we can do that.
One of the biggest issues when I was growing up was fluoridation of water.
Oh yeah,
and you had all these.
I remember the posters of kids with rotten teeth.
Yes,
about how this was what it was going to do to your.
That's
teeth.
right. It was a communist plot. I mean, it was like a subversive plot. Because look what fluorine does to this worm. You can kill it with a lot of it. Hydrofluoric acid will eat through glass. You're going to put that in our water? Wait a minute, it's already there. That's how we discovered it. And so that it blew over once everybody's dental plans started to go begging for use.
Well, and I think it also helped that that viewpoint was very, very powerfully satirized in a major motion picture that was quite popular.
Yeah, that's true. Well, we still depend on satire for diffusing all these things.
But there are major motion pictures that indict the pharmaceutical companies as being evil and only wanting to make money and hurt people. And it's there's no basis for a lot of that. They do want to make money, of course, but hurting people doesn't
make money.
No, that's right. So a college education should force you into rational behavior, but it often doesn't.
So, Timothy, I hope we've solved the problem for you.
Yes, I think that pretty much wraps up the whole
You're suffering from our own dilemma here, kiddo. Join the group.
There's no answer, but it's a great topic. You could talk a long time.
Some investigators refuse to talk to the press because of that.
Exactly, sure.
Which I think is the wrong approach.
I do too. Of course it is the wrong approach. Of course it is.
I have no problem, as many of us do, but many people who make the big discoveries don't. So that's a big problem.
That's right.
Anyway, Timothy, hope you like that. Thanks for listening. Next one is from Ilya, who writes, hey, look at this. Hello, Vincent, Alan, Rich, and hopefully Dickson.
Okay.
She knows exactly how to address us.
You got it.
Oh, shucks.
I write to tell you again of how fascinating and useful I find TWiV, your ability of being descriptive and as simple as possible while talking about serious questions in virology attracts people and makes them wait passionately for each new episode.
My goodness. Interestingly,
this TWiV phenomenon spread far beyond virologists. You are great. No, we're not great. No.
Is that G-R-E-A-T or G-R-A-T-E?
Grading. Yeah, we're grading.
We're grading.
Recently, I had a talk with a non-virologist on how could a non-living object multiply and influence a cell so much. Another chapter of chicken and egg question in virology. Successfully, I managed to find a decent model for explaining it to him. Now I want to share it with you. Imagine a fine Swiss-made mechanic watch. Each gear wheel is in its place and everything works fine. Suddenly, one tiny tooth slips through a chink inside the watch case and gets stuck between two gear wheels. The watch is still ticking, but something goes wrong from now on. One by one, teeth start to break from stalled gear wheels, damaging working ones. Now the watch case breaks Now the watch case breaks open, exploding lots of tiny teeth to other watches.
Oh my goodness.
That's great.
I can see that movie.
Yeah, that's good.
Maybe it's a bit too mechanistic, but descriptive. It worked.
No,
Great.
it's a good analogy. Very good. Thought process.
Listeners, vote for more TWiP campaign.
Hey.
All right. Next one's from Arson. Hey, TWiV88 gave me some pretty nourishing food for thoughts. I'd like to thank you for bringing my attention to the article on gene therapy being a potential cure for HIV. I'll translate it and publish it in my Ukrainian blog on advances in medicine. So he has a blog which is in Ukrainian. I can't
read it, but the pictures are great.
Cool pictures.
I don't know where he got these pictures, but they're cool.
Uh-huh.
Also, I have found the article on reconstruction of an ancient human retrovirus fascinating. It would be interesting to conduct a different experiment. How about removing from a human embryo the traces of recent, since apes, retrovirus infections and observe if there are any noticeable differences? The experiment probably wouldn't be conducted because of the ethical considerations. However, it would be just fascinating to find out if some of our superior human intellect was originated by retroviruses.
You could do it on a chimp.
Well, you got a big job ahead of you because there's a lot of those.
I'll say.
Yes.
Well, you could, in theory, do it in mice, but we don't know how to actually remove them. And so it's a difficult experiment. And then once you do in a mouse, what do you look for, right?
Right.
So what is that fish? It's the Fugi or Fuji fish?
Fugu fish.
Fugu.
That has a really small genome, right?
Yes.
Yeah.
Does that thing have any endogenous retroviruses?
Good question.
We talked about this.
We talked about this. I think it turned out that there were, but
yeah.
they're very, very few.
Yeah, there are. I think everything's got an endogenous.
So maybe if we took out all our retroviruses, we'd turn into a fugu fish.
We'd become toxic, that's for sure.
It's an interesting experiment, though. The next one's from Ed. I came across a book the other day that scratches the same itch as listening to TWiV and TWiP. It's called Genes, Germs, and Civilization. Isn't it by Jared
Diamond?
That was Guns, Germs, and Steel.
No, that was
Oh, okay.
Not by Jared Diamond.
Sorry. Can someone search on that?
I'm doing it now.
I'm only a little into the book, but so far it's covered some interesting links between the origins of diseases, measles, malaria, versus the human population density needed to keep the infection from dying out. I wonder if you've read it and what your opinion on it is. I haven't read it.
It's by David Clark, Germs, Genes, and Civilization, How Epidemics Shaped Who We Are Today. $16.49. Kindle edition is available.
What year was that?
That is scrolling. First edition, May 2010.
Really?
So it just came out.
Brand new.
I don't know. I have to look at it. I will.
That one passed under my radar screen.
Have you guys read Guns, Germs, and Steel?
I have not.
I have not.
Yeah.
I've tried to read it. I read his book Collapse.
So I need to read both of these because they sound very interesting.
They are interesting.
He continues, I've been listening to TWiV since your first appearance on Futures in Biotech early last year. I'm an electrical engineer, and as others have mentioned in letters you've read on the air, there's something compelling about viruses and their programming. The question of whether computer programs can mutate and develop successful new features came up on one of your recent shows. The biological system has an advantage over present-day computers in that each cell is equivalent to a computer. In an infected individual, you essentially have billions of parallel computers, each producing occasional random variations, and billions more uninfected computers, each testing the outcome. Someday we may have that many computers, but so far it's a math thought experiment.
That's a really nice way of looking at it. That's a new twist on the problem that we've discussed before. I like that.
Yep.
I think there have been some artificial intelligence experiments that have sort of done super simplified versions of this, where you you have data structures that you don't have millions of computers. You have millions of these copies of these data structures that randomly change themselves. And then you compare them to some competitive activity, artificial activity that would give them advantages, and sort of try to simulate evolution that way. But he makes a really good point that the total processing power in a computer is actually just not even there.
Yeah, I once watched a demonstration by Gerald Edelman, who got involved with learning behavior and invented a 1,000,000 electrode machine that could track a ping pong ball with another ping pong ball, and actually got the second ping pong ball to actually follow the first one by learning the behavior of the first ping pong ball. So it was quite an interesting simplified version of machine learning. And I would hate to think that we're reducible to something like that, but with trillions of neuronal connections, maybe it's just a matter of mathematics at that point. I don't know.
Well, we're probably simpler than we like to believe.
I certainly am. I don't know about you guys, but when I got to the subject of virology in college, I stopped. That's why I'm trying to pick it up again.
Well, now you're working on it again.
Well, I am, because I'm now I'm intrigued as you guys are.
Yeah. Next one's from Samuel. I love TWiV, and I hope it becomes even more popular / famous. I read H1N1 Lessons Learned in April's CIDRAP. CIDRAP, Center for... I can't remember what CIDRAP stands for. Let's look it up. I look at it often, CIDRAP. The Center for Infectious Disease Research and Policy at the University of Minnesota.
Uh-huh.
An article by Robert Roos in which you, Vincent, were interviewed, and I was surprised that there is not an agreement among scientists on what to expect from this upcoming flu season. Why is it that some virologists have doubts that the 2009 H1N1 will replace other subtypes as the major seasonal flu? Is it mainly because the cycling strains of the 1977 H1N1 epidemic and possible lab accident muddle things so that we have no analogous history to compare to the current situation? I don't want to sound like a pushy reporter, but I would be interested if anyone would like to place a bet on what we will see from H1N1 this autumn.
Bets.
Can there be only one serotype that carries the day and becomes the major seasonal flu? I ask because in 2009, H1N1 seemed to have usurped the previous flu. What is the significance of serotype? If the 2009 H1N1 and the 1957 H1N1 have the same hemagglutinin and neuraminidase proteins, does that mean they have the same antigens? Do they elicit the same immune response? Thanks for bearing with me through all these questions.
They remain questions.
Well, let me just say that it's unfortunate that they're all H1N1 because they're all very different. The 2009 H1N1 was very different from the previous seasonal H1N1, which is very different from the 1957.
It's almost like they're going to have to come up with a revised nomenclature to deal with this. The serotyping nomenclature used to make sense, but now in the genomics age, it's becoming almost archaic.
Yeah. So to the public, it seems like they're the same virus, right? They're H1N1, but they're not.
Same, but it isn't.
So in fact, if you were infected with a 1957 H1N1, you're not going to be protected against the 2009 H1N1. They're very different.
Although there was partial protection in older people, right?
Yeah, so if you were infected with H1N1s from before mid-50s, there's some partial. In fact, if you had the 1918 and survived, you were quite well protected against the 2009.
If you were still alive.
If you're still alive. No, if you're dead, you were not protected.
No.
Right.
Or you were.
No, actually,
depending on your perspective.
Yes. If you're dead, then your condition has stabilized.
That's right.
So they do not have the same antigens, and they do not elicit the same immune response. As for this multiple circulation thing, this is tough because for many years only one seasonal strain circulated in any given place. And that changed in 77 when this H1N1 came back. So who knows now what's going to happen. So now we have a little bit of benefit of hindsight. This is an old letter, I think. It looks like the old seasonal H1N1 is gone. And we're left with the 2009 H1N1 and a little bit of H3N2 still circulating.
Yeah.
I don't know why they co-circulate. I don't think you can predict what's going to happen. No one can. because you just don't know how they are going to interact with the immune population. That's basically it. You can't predict it.
Initially,
we can't predict the. Oh, sorry. Go ahead.
Initially, the 2009 H1N1 really kind of took over. It looked to me like it was going to displace everything, but now it's sort of re-equilibrated with the old seasonal strains. So,
yeah, if you look at the numbers,
that.
if you go to CDC and look at the numbers, I mean, most of the isolates are still 2009 H1N1. There are an equal number of influenza B, and then there's some H3N2s scattered throughout. So I would guess that the H3N2s are going to go away at some point.
Well, and we can't. One of the major reasons we can't predict how this is going to happen is we don't have perfect and instantaneous data. So you get influenza. I mean, most of these cases, if they're even reported, most of them are not going to be reported. The ones that are reported are mostly reported as influenza-like illness.
Right.
And so the CDC ends up tracking cases of ILI, and you don't know what strain that is unless you go and test. And who's going to pay for all this testing? And during the H1N1 pandemic, there was a lot of testing going on because it was very important to track. Since we had separate vaccines, it was important to track which one was on top. But if you know what your three most likely dominant strains are, and as long as one of those seems to be emerging as one of the vaccine, the strains that's in the vaccine seems to be emerging as a dominant one, then you just vaccinate.
That's why we need a universal vaccine, so you don't have to worry about
it.
Then
we don't even have to have this conversation.
No, you get it when you're young, and that's it. Protects you for life.
Right.
So, and making these predictions, I really think we just really don't know enough about the viruses. As much as we know, the factors that make one virus dominant in the population are many, I'm sure, having to do with transmission and replication and immunogenicity and all the rest of that. And we just don't know enough about it. That was one of the interesting things about resurrecting the 1918 influenza and asking about its pathogenicity and what the virulence factors were. And I think one of the lessons that came home from that was that it's a constellation of effects. It's multifactorial, and we don't even understand that. So it's a complicated problem, and the predictions are going to be far from perfect or impossible.
And the other thing that strikes me here is that we began a conversation a long time ago in this series about medical ecology. The sense of keeping track of all the other hosts as their wildlife habitats are being shrunken down by encroachment and increased human populations. These bird migration routes might be changing, in fact, and because of climate change issues, that might be true also. So the distribution of the birds and the distribution of the viruses that they carry may be also in flux to a point where we're not able to keep track of this any longer. The reason why I wanted to say that was because there's a wonderful show coming up on Sunday on the Discovery Channel called Migrations.
Oh, cool.
And you guys should really pay attention to that, I think. It's going to cover all kinds of animals, but I'm sure that birds will figure in really into this. And it brings me up to my pick of the week, too, by the way. So
good.
When you get to that, just remind me again.
Just a last thing here on this discussion. If the CDC has just started publishing its weekly surveillance of the flu isolates in the U.S.
Yep.
So we have the first week at flu. It's cdc.gov/flu/weekly. And for week 41, we're in the 41st week, or just past it, ending October 16th. 2,571 specimens tested, 3.6% positive for influenza. And of those, 16% were the 2009 H1N1, 26% H3N2, so actually a little more than I said, 57% not typed.
Right.
And also 26% B. So H3 is still there. I'm wrong. I thought it was going away, but
nothing ever goes away, right?
Nothing ever goes away. Neither does TWiV.
No. Nor should it.
Let's do some picks of the week. Now, Dickson, you said you had a pick of the week.
I do. In fact, it's called Winged Migration. And I don't know if we've ever picked that one before, but let's pick it again if we haven't. Winged Migration is a wonderful film made by French film producers who also filmed another wonderful show called Microcosmos.
It's going to be on Discovery.
No, this is an older film, but it's a fabulous film because it actually It puts you into the bird's brain and lets you look out through its eyes at its migration routes. I don't know. Any of you guys ever seen this one?
No.
Is this the one where they actually followed birds around with a ultralight?
That's correct.
Okay.
That is exactly correct. It's a fabulous, fabulous movie, and it's essentially it's done in silent movie mode with music in the background. And it's absolutely wonderful and stunning, and it well, as long as we're discussing influenza, we might as well know where the birds go too, right? And so
sure.
this
I'm not sure I've seen this exact one. I'm gonna have to put it on my Netflix.
You'll love it. You'll love it, Rich. This is your kids will love this one too.
Great.
Cool. Yeah, here it is on the Internet Movie Database. Documentary on the migratory patterns of birds shot over the course of 3 years on 7 continents. How many continents are there, Dickson?
Well, I guess there are 7, aren't there? I would dare to guess that there are 7, but
for now, anyway.
That's right. That's right.
Does Antarctica make 8? Or
now remember, there used to just be 1. So
yes.
What was that called, Dickson?
That was called Pangea.
So, what about what Alan just asked you? Does
Antarctica, is that the 8th continent or is that the 7th?
I think
that's the 7th one because Greenland is considered an island, and so is Australia. So
we're revealing the boundaries of our knowledge here.
Exactly. Let's not reveal
We're
too
not
much.
This is not TWiV.
That's right.
This is not TWiV.
No, this is TWiPs in the worst sense of the word. Twerps.
Ooh,
Alan, you're next.
Well, my pick is a website called WebSDR. And this is a site that has a listing of web-accessible shortwave receivers located around the world.
Be darned.
So these are radios, and what's really cool about these is they're not. People have been plugging radio receivers into the internet for a number of years now, but the usual arrangement is that they'll be listening to one frequency, and you can go to the website and you can, if you are the only one there, you can change the frequency to something else. And if somebody else shows up and changes it, then the station changes again. The neat thing about this is that you can have multiple users listening simultaneously to different streams because they're all software-defined radios.
Neat.
And it gives you they give you a really cool display when you go to one of these things. It's called a waterfall display, so you actually see the signals that are on an entire band.
Yep.
And it's aimed mostly at ham radio operators, so the bands for most of them are the ham radio bands. But it's kind of cool to listen to, even if you're maybe cool to listen to, even if you're not a ham. It's mostly people talking about radios, but you can tune into these things, and they're in different locations around the world.
Do you do this, Alan? Do you tune in?
I had just discovered this the other day.
I looked at this. I looked for Alan on this. I didn't see him.
I am not on the HF frequencies at the moment. I haven't gotten my antenna back up since moving last year, but yeah.
You have gear, right?
I have some gear. I'll probably acquire some more, and yeah.
People who have gear always acquire more.
Yes, exactly. Gear multiplies.
Gearheads. Yeah, that's pretty cool. I have never done shortwave. Have you, Rich?
No.
So I could go on here, click on one of these sites, and talk to these guys, right?
You can't talk through it unless you're. Well, these are set up so that they're receive only.
So you listen.
Yeah, because if they allowed you to transmit and you're not a licensed ham radio operator, that would violate the law.
I see.
There are setups with some of these where people remotely control a complete transceiver over the internet.
So there's this one guy at the Netherlands. One guy in the Netherlands has got 1, 2, 3, 4, 5, 6, 7, 8, 9 frequencies going. Is that
9 bands? Yes.
9 bands. Wow.
Yeah. So Alan will be doing that one day.
Yeah.
He'll be doing This Week in Ham Radio.
I think there are already about 50 of those on the podcasting circuit.
Is that right? Yeah.
Yeah, hams were, as you'd expect, they were among the first in podcasting.
They like to talk, right?
Exactly. Like to talk, and they've got the gear.
Cool. Rich.
So we talked about this before, but it wasn't a pick. I looked, and I would like to replug this one. Personalgenomes.org. This is George Church's project at Harvard University to sequence, what is it? The ultimate goal is something like 100,000 human genomes.
Yeah.
And make both the genomic data and the medical records of the individuals who've been sequenced public along with it. as a public database resource for understanding genetics and in particular how it relates to human disease. And I think this is a fascinating project, and in fact I'm signed up. Me and my wife are signed up, and we have identical twin daughters.
Oh, wow.
Which I thought these guys would be really interested in that, and we're going to get my daughters signed up as well, and get our genome sequenced and see what comes out. I've even got a little number and everything. You signed up, didn't you, Vincent?
I did. I went through the second phase, which is to take a bunch of online tests.
Right, I did that.
And now I think in the next week or so, they're announcing the next 10,000 people that they're going to.
Wow.
So at some point we have to go up to Boston and give them some cheek swabs and.
Oh, you have to give them a limb. Forget the cheek swab.
Well, they're going to start cell lines, among other things. Yeah. I want my cell lines.
Yeah,
I want to grow my cells and infect them.
Yep, me too. I want to do plaque assays with my
cells. You guys, jeez.
Well, we could do this
Hamish,
on our own.
I remember Hamish said that a number of years ago he had a tumor removed and had somebody waiting with dry ice outside the OR, one of his lab mates, to take a piece of the biopsy and see if they could culture it and develop a cell line, because he thought it would be really cool to have hey-yo cells.
Yeah.
That's right.
Yeah, they don't.
At any rate, I think this is a wonderful project, and I'd like to plug it.
Yeah, go and sign up.
Collect some DNA for these people.
They don't actually take a limb; they just take blood and cheek cells. Yeah,
right. I'd love to know how much part of you are Neanderthal.
Right.
Well, I could tell you how much you are.
My wife can tell you how much I am. I'm sure.
And as a footnote to this, I know we have a lot of IT and computer folks listening. I see at the bottom that they are personalgenomes.org is looking for people who develop Rails applications.
Yeah.
Ruby on Rails.
Definitely, yeah.
Oh, and the other thing I found while I was just looking through the website is that there's just yesterday. George Church gave a TEDMed talk. And he's out at the TED conference somewhere on the West Coast. And that presumably will go public at some point.
So they've done the first 1,000. They have the PGP-1K. Wow. You can see them here, and they're listed, and you can click on any one of them.
Holy cow.
And see the information and what kinds of diseases they have found. So they have, like, this person has mild allergies to a variety of things. He or she's gotten vaccines, so you could sort of correlate some of the medical history with the sequence.
Now, what's going to happen when they apply for health insurance?
Well, this is
just putting that idea out there.
No, this is a significant question. There's a big, long consent form that you go through. Part of the registration process is to assure them that you're aware of all of the potential negative consequences of doing this. Okay, right. And a big aspect of this is the ethics involved. They're researching the ethics at the same time.
The first 10 actually have their pictures here and names.
Quite amazing.
George is one of them. George Church was a big guy in developing the earliest sequencing technology, and I remember him very well because I was doing that years ago.
So does Leroy Hood have his done in Hunkapiller also?
I'm sure.
Very cool thing.
Very interesting to do. Great. Yeah, there's so many cool things about the genome. I'd suggest people go over to Futures in Biotech. There was an episode with George Church. Really interesting. Very smart guy. Well, my pick is Ex Vivo, a site where you can see amazing scientific animations, and this was suggested by a fellow on Twitter, JHARSEM. I'd known about this for a while, but never made it a pick, although we've selected some movies now and then, and it is just gorgeous.
It is gorgeous. I was looking at some of these before the show. They got a couple of virus ones. What I like best is in the inner life of the cell, they've got, actin motor molecules.
Yep.
Right.
These things walking along
walking.
microtubules and carrying big balls. And, that's probably not far off what they look like.
No, these are beautiful.
It's just wild.
I show this to my class. That one, the walking one, and some
Yeah.
other virus entry ones. There's a whole variety of stuff here.
Really good.
And if you don't know about it, check it out. It's just beautiful.
This stuff is gorgeous.
I have only one objection to this,
Yeah.
and that is that my sense is that the inside of the cell is like probably a gel.
Yes.
It's really dense.
Or a liquid
These are
crystal, at least a liquid crystal.
Yeah, these are all drawn as if it's like underwater. Or
Yeah, there's some
something.
space.
I
You're
don't
right.
know how else they can do it.
Oh, Rich, you're right.
But that element of it, I don't think is quite right. But the animations are fantastic.
Yeah, well, I guess
Yeah,
that-
you have to warp reality in order to show
Yeah.
these things. It would also be very dark inside a cell.
Yeah.
and there
They
would
got
be
little flashlights,
probably not be as colorful, but to make it clearer, yeah, it's an education.
If everything was labeled with fluorescent green protein, it wouldn't be so bad.
It's edutainment.
It's fun to look at this like the inner life of a cell, and you look at something, you say,
Here, here.
and then all of a sudden it clicks in, oh, that's messenger RNA.
It's very cool. Yeah, it's just beautiful. So,
Okay.
thanks, Jarsim, for reminding us, and there you have it. And there you have another TWiV. TWiV you can find at iTunes, the Zune Marketplace. You can listen to it on your iPhone or iPad with the Micro World app. Or you can go to twiv.tv, download it, or play it right there. And there you can find show notes, transcripts, and questions, as well as picks of the week. And speaking of questions, tell us what you'd like to know. TWiV at twiv.tv. Dickson, good to see you again twice in a row.
I could even try for 3.
Always good to have you.
Love being here.
Verticalfarm.org. And I have a link here for an NPR article about the vertical farm, which I'm going to supply.
Good.
As well as a link to a YouTube video of you being silly about vertical farms.
This is true.
The Leaning Tower of Pizza.
No, I rejected that, and then it fell over.
We'll put those 2 links in the show notes.
Okay, good.
You can find Alan Dove at alandove.com. Good to have you.
Yes. Good to be here.
Always Western Massachusetts, the heart of this country. Or I should say the origins.
Something like that, yes.
Yeah. And Rich Condit down in Florida where the football isn't so good, University of Florida.
We'll be back.
That's right.
We'll be back.
That's right.
Thanks
Great
to
to be here.
everyone. Always good. I'm Vincent Racaniello, and I'm right here next
You
to
are.
Dickson
That's right.
on the Skype. You can check me out at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
I love Schmutzdecke. Schmutzdecke. Ja. Ich liebe Schmutzdecke. Ich bin Schmutzdecke. We will become Schmutzdecke, believe me.